Maharashtra University. He has supervised 60 students, PhD students, scholars, and currently 8 students are enrolled under the supervision. He has guided several MSc, M.Pharma students for dissertation work. More than 35 scholars received prestigious overseas fellowships and research publications, and their Honorable Sir has more than 205 national, international publications in peer-reviewed journals, and their Sir has been invited as a speaker and as chaired the session nearly 50 national and international conferences. And he has worked as a project investigator in number of projects funded by USA, DST, UGC, and CSIR, etc. I think around the rupees worth 25 crores. This is the very first. He has visited many countries as a scientific purpose. He held various important posts as President, Indian Society of Chemists and Biologists, and currently they are Chairman, Gujarat Vidyapith University. Thanks, sir. I'm heartily welcome to you. You have delivered to your speech. Thanks.
Thank you very much for nice introduction to me.
Thank you, sir.
I am really thankful to our Center for Refresher Course at Gujarat University, and also Professor Wasawa for inviting me on this occasion.
Welcome, sir.
Yeah. So, in fact, what has happened that during this COVID-19 days, 19 time, we had enough time to go through what is happening with the COVID-19 drug research. And though you know that by training and by profession and by all experience, I am a hardcore synthetic organic chemist, converted into medicinal chemist later on, and working in 3 interesting area: anti-cancer, antiviral, as well as anti-tubercular. So majority of my work in last 30 years was pertaining to drug discovery in these 3 typical area, and some portion of analytical chemistry, and some portion of newly now field of interest of many people, that is the deuterated drug molecules. So many projects we have completed at Saurashtra University, a very good research center which was supporting more than 50 universities and 250 research scholars all this year. I was a part of founder of that center, and later on this center has worked very well for giving analytical support to researchers in Gujarat and rest of the India. So what I was thinking during these COVID days, and every day newspaper was coming with when the vaccine will be developed and how vaccine will come. So I diverted my interest, and I had become a student like all what we call it lifetime student, and I started learning about the vaccine development and what is the updated vaccine development in last 6 months and earlier, and how the COVID vaccine development works. So today's my talk is not the synthetic organic chemistry, not the reaction mechanism, not the HIV or anti-tubercular or cancer, on which I have given several hundred lectures, but I will focus only on the latest update of COVID, and I am thankful to Central University, Gujarat, who pursued me to speak on this occasion. So it is a latest update regarding the COVID-19. Friends, we know that there are two types of development which are taking place in the COVID drug research, and we are aware about that when initial stage everybody was discovering the molecule, or they were repurposing the molecule, the repurposing of old antiviral drug or old anti-malarial drug like hydroxychloroquine, and there was a worldwide notion that the many more molecules have come up and these molecules can be utilized. And so hydroxychloroquine, remdesivir, and later on favipiravir, which was used for antiviral purpose for some other use, and we found that the virus, a very notorious virus, can be treated by these drugs. But ultimately, when worldwide results are coming, these molecules have not shown very good performance, or what we call it as a blockbuster drug, which can resolve at a time with a single drug all the problems. And therefore, the problem has become very tricky, because region-wise, and if you look at the world map, the COVID problem and the death due to COVID virus is differently classified. So in some cases, it is seen that it is the collapse of the lung function and the lungs were totally damaged and the death is due to lungs. In some cases, to our surprise, it has gone to liver. In some cases, it was found that it can cross and it will reach to the brain, and due to cerebral stroke, the people have died. So cardiovascular, and the fourth problem is comorbidity. And it is said that old age people with the phenomena with what we call it diabetes and cardiovascular function and other ailment is increasing the death toll of the COVID patients. So under this background, it was worthwhile to understand what is this virus, how it works, and we know that COVID-19 is nothing but it is one of the modified virus which we have faced earlier in case of the SARS virus. So it is a type of SARS virus, and where we can study it and understand that how this COVID-19 works. So I think that I will go to a PowerPoint presentation which will explain particularly regarding this virus, and we can know about the basic facts of this virus. So let us talk about certain basic fact about the COVID-19. As it is known, as I told you, it is known as the SARS-CoV-2, and we can consider it as a single-stranded RNA genome which has 34 kilobases and having a nucleocapsid of helical symmetry. The SARS-CoV-2 genome is 80% identical to the SARS-CoV and 96% to the bat COVID, and that's why we know that everybody is talking about the bat. So it is 90% similar to base COVID RA-TG13. The integrity of the SARS-CoV particle is maintained by 4 proteins. Now let us look at these 4 proteins. The first is known as the S protein, which is a spike protein, which is enabling the attachment of the virus to the host cell, and this S protein followed by the membrane fusion. So it is basically promoting the entry of the SARS-CoV into the host cells. The second is the abundant M protein, and this M protein, you can see that there are abundant M protein membrane that maintain the membrane integrity of the viral particle. Then comes the envelope, and that is known as E protein. It is the smallest protein and plays a structural role and helps in assembly and budding. And fourth is the nucleocapsid, N protein, whose basic role is to bind to the SARS-CoV RNA, as it is a single-strand RNA, and supports the nucleocapsid formation. So this is how we can see it. And the next important thing is how it looks like. So you can see that this is a presentation drawing which is showing us membrane protein, that is glycoprotein. It is showing us the envelope protein, a small one. It is showing spike protein, and on other side there is a nucleocapsid protein, RNA and nucleocapsid protein, which you can see, look at here. Now this is the process of COVID-2 and ACE2 binding. Now any ACE2 receptor, when binds with this, then the first step starts. The second step is the proteolytic cleavage, followed by the fusion, and you can just see that it is the genomic RNA positive, and the viral RNA is released during this process, and nucleoprotein is uncoating. So you can just see that RNA dependent RNA polymerase, and this RNA polymerase with the enzyme replicase, the another step starts translation and replication, and then transcription step and RNA replication and packaging. Then the role of mRNA, and six is the translation, and seventh is the assembly and budding, and next is the exocytosis, and ninth is the viral release. So this is how the entire process is going on. Hello. So what we have understood on this, that how this ACE and receptor binding domain are closely connected. You can just see that this is the close connection, and this is how the entire process is going on. So this is the SARS-CoV life cycle, and viral S protein binds to ACE receptor, and following this entry, there is a proteolytic cleavage of the virus envelope, ensuing the release of genomic RNA into the cytoplasm, and the whole process we can read on this slide. And there is a very recent work which was reported by Chauhan and other in the SES Nano. The next is again a important function where healthy and dysfunctional functional immune response during the infection. What happens during the infection? So what happens that the virus infected cell, it undergoes a pyroptosis and generates the molecule, including the damage associated molecular pattern, nucleic acid, ASC oligomers and ATP, and it will ultimately trigger the two type of cells which are in the neighbor thing, epithelial as well as endothelial and macrophages, and the proteins which are present, cytokines and chemokines, pro-inflammatory proteins, they are released there and migrate the T cells, and this is what is happening in this. And what happens that the monocytes, T cells and macrophages will go to the infection site, and a loop of pro-inflammatory feedback is started by IFNY mediated elimination of the infected cell. It will neutralize the antibody mediated, which is produced by the beta cells, and viral inactivation. And the whole process biochemically, the dysfunction of immune response can be seen on these three macrophage, T cells and monocytes. And what we are facing is the cytokine storm due to IL-6, IP-10, IFNY, IL-2, so interleukin 6, 2, 10 and TNF. They are playing their role, and ultimately we can say that the in the lungs, the alveoli macrophage clears up the neutralized virus, neutralizing antibody binds and inactivates the virus, and macrophage recognized and phagocytosis apoptotic cells, and then there is no viral release further, and there is a role of CD4 cell, CD4. So T cells mediated efficient immune response is obtained as a healthy immune response, and infected cells are rapidly cleared. Virus inactivated by neutralizing the antibodies as mentioned in above mechanism, and minimum inflation and lung damage takes place. So this is how what is happening. Just before few seconds, I told about the cytokine storm. Now, what is actually happening? It is the disturbance of the total cholesterol biosynthesis and the sterol regulatory element binding protein, which is activated by the cytokine or pathogen, which is known as virus or bacteria. It is confirmed that it is very recently confirmed that SREP BP2 induced cholesterol biosynthesis suppression by cystine-1 and by this expression, and the inflammatory responses of this was upregulated in COVID-19 patients who are in ICU. So this is the how signal transduction pathway is observed during this all responses, and how this COVID-19 infection leads to the cytokine storm, which is responsible for the entire lung damage. And this happens particularly within few hours, overnight, and ultimately the patient may die due to this. So the therapeutic drugs which are given to the patients are related, which can avoid this cytokine storm, and it can help the patients to recover from this by the viral inactivation. The next important aspect is the role of CD4 and CD8 cells and how the lymphopenia, T cell activation, lymphocyte dysfunction take place and how immunopathology of COVID-19 works. As I have told you that interleukin plays a very important role and the antibody are increased. And on other side, you can see the granulocytes and monocytes and abnormalities on this, and where neutrophils and eosinophils, this is basophils are down, and you can see the dysfunction, how it is taking place. So you can see that immune pattern of COVID-19, it will include lymphopenia step, lymphocyte activation and dysfunction, and abnormality of granulocytes, increased production of cytokines, and then it will lead to the increased antibody. Especially lymphopenia is a key feature of the patients with COVID-19, especially in severe cases. While CD69, 38, 44 are highly expressed on CD4 and CD8 T cells of the patients, and viral specific T cells from severe cases exhibit a central memory phenotype with high level of IFN-Y and TNF-alpha and interleukin-2. However, lymphocyte shows an exhaustion phenotype with the programmed cell death protein, that is PD-1. So it is a apoptosis type of phenomena, programmed cell death. The next important part of this is potential mechanism of SARS-CoV-induced immunopathy. Again, the first step is depletion and exhaustion of lymphocytes. The spike protein is here. ACE2 is the binding. This is the T cells. This is the impaired T cells. Then T cells depletion and exhaustion takes place, and role of interleukins. Then viral infection will damage the spleen and other part, and lymph node, which is where we can say that it's a lymphopenia phenomena, and then T cells is impaired and dysfunction. The second is increase of neutrophils, where lymphopenia, microbial infection takes place, activation and recruitment of neutrophils takes place, and this both phenomena will subsequently show us the potential mechanism of SARS-induced immunopathy, and the entire description of is given here. Due to paucity of time, we are not going into the detail, but you can just understand that the three important processes in this interrelated, and the cell virus entry, disease progress, and poor outcome, which comes due to antibody dependent enhancement. The next is the clinical implication of SARS-CoV-induced immunopathy. The patients with COVID-19 and who are present with the lymphopenia are more prone to infection by the microbe, which leads to disease progression, and ultimately it is increased severity. We know that many patients who are COVID positive, after some time, due to their immunopathology and their inner immunity, they are coming back after some time, and mild to moderate to severe cases, they are treated by the different group of drugs, but not the vaccines as vaccines are not discovered so far, and therefore it is a big problem that a group or bunch of drug which includes steroids, which includes vitamins, which includes antibiotic, antibacterial drug, and some of the drugs which are known antiviral drugs and used for antiviral, they are to be used for a composite or integrated treatment to the patients. So that is how we know that in COVID hospitals, there are variety of treatment which are given, and every hospitals have come out with their own protocols and treatment, and many people are recovering from this problem. So here this is to show the clinical implications of the SARS-CoV-2, and what is happening here, that cytokine storm will also initiate inflammatory induce multiple organ dysfunction. We have already discussed that it will include the lung injury that can lead to acute respiratory distress syndrome and total respiratory failure. As I have told you, the liver injury with ALT and AST and YGT upregulation. It will also lead to the kidney injury with the include urea and creatinine level, and heart injury with increased creatinine. The recent development to the brain damage and stroke is not covered in this research paper, but when we are discussing as a whole, we have to consider the effect on other organs and in our cerebral system also, that how the SARS-CoV responses to the various organs. So you can just see that the effect of elevated cytokine production on severe syndrome, cytokine storm, lead to multiple organ dysfunction, lung, liver, kidney, and heart. And this is the lymphopenia where microbial infections is also increased. The next is to give you an idea that what are the chemotherapeutic and vaccine development for SARS. And there are few enzymes which are responsible, and where we can think on how to work different strategies for this target identification to hold the pathogenesis of viral infection. It is most important, and then viral protease, host cell produced protease, RNA polymerase, and interaction site of viral S protein with host receptor AC2O, ACT2 are among, these are the major target which can use for the repurposing already existing antiviral molecules and new small molecules which are under development. We know that in last few years, the number of new drugs which have come out for different diseases and the number of the old drugs which were repurposed, small molecules are repurposed, other molecules are also repurposed, macromolecules are repurposed, and repurposing the old molecules, safe molecules, safety, efficacy, and stability and shelf-life period are important in drug discovery. And many things which has happened all these years is the repurposing of the molecules. So in next few slides, we will take on that what are the molecules which are very important, particularly the existing antiviral molecule used for the different purpose. And you can see that the number of molecules which are mentioned here, name of the molecules is structures, are mentioned here, and umifenavir, EIDD2801 molecule, remdesivir molecule, then galidesivir molecule, then favipiravir, then thalidomide. Thalidomide was originally a anti-cancer compound. Tenofovir, baricitinib, ruxolitinib, which is anti-cancer and antiviral, chemostat, hydroxychloroquine, chloroquine itself, lopinavir, ritonavir. These are all antiviral drugs which are basically used as a repurposing, particularly for the COVID-19 therapeutics, and various laboratories, doctors and physicians, they are trying on these molecules, they are collecting this data, and in many cases, the mild to moderate, the benefits are observed by any of the single molecules or when it is used as a collective with the other molecules. So this is an exceptional slide which I want to show all of you, because majority of the participants in this webinar are from the chemistry side, and they can understand that we have a quinoline molecules, we have a, other heterocyclic molecules, pyrrole molecules, we have other fuse systems and nucleotype molecules, and then we have a single ring system molecules like favipiravir. And incidentally, I am very happy to inform you that the favipiravir is independently and indigenously prepared by some of our students for the Indian and global market, and they were one of the first people to manufacture and to prepare this molecule in India. So this is how the work is very much done within India. Other companies have also worked on many molecules, old molecules, repurposed molecules, and it is now marketed and it is accepted by our drugs department and our what we call it licensing authorities. I'm coming back to my talk to vaccine development, and when we do the significant genomic match of this virus with other coronavirus, then it will help the vaccine developer to facilitate the designs toward the most promising vaccine candidate. Now, in next couple of slides, what is the present status of this? And I can say that the vaccine development of this candidate is relying on not only one platform. There are several high-tech platforms, including the attenuated and inactivated virus also, replicating and non-replicating viral vectors, DNA and mRNA, virus-like particles, and recombinant protein-based approaches also. So, when people are not aware about the vaccine, we have to understand that the chemistry teachers and chemistry researchers that there are not a single platform. There are several platforms on which world level people are working. The people who are working in Boston, who are working in Oxford, who are working in Europe, who are working in India, they are using different platform for this viral vaccine. Now, let us understand that what are the different vaccine platforms. So you can see that there are classical platforms and there are next generation platforms. On your left hand side, there is a schematic representation of classical platform, which include wall activated, inactivated virus. For example, polio vaccine was based on that. Then MMR vaccine, live attenuated virus is used. Then protein subunit is used, and in influenza virus vaccine, we are using that. Then virus-like particles, they are used in human papilloma virus vaccines, and it is at a preclinical stage. So this is the classical platform, and what are the next generation platforms? So viral vector, for example, Ebola vaccine, DNA platform, which is not currently licensed to anybody, but INO 4800, it is in the phase 1 clinical trial. Then RNA, not currently licensed, but mRNA 1273, BNT 162, they are in the phase 1 and phase 2 clinical trials. Next comes in the antigen presenting cells. And you can just see that LV, SME, NPDC, and that is the COVID-19 small a, capital APC, it is in the phase 1 and 2. So these are the various virus platforms where viral vector, DNA, RNA, and antigen presenting cells are used. So if we look at the latest information and literature, we can say that the LAV vaccines are well known. It is the most immunogenic vaccine that do not require adjuvant to gain optimal response due to its effectiveness to provoke immunomimic to the natural infection. So marketed examples of LAV is in mumps, rubella, measles, and varicella vaccines. So it is a very profound and well received vaccines, and Indian company Serum Institute of India, frequently its managing director is coming in television. In collaboration with Codagenix Biotech, they are working on this vaccine, and we are awaiting the good results from that. The second is inactivated wall virus vaccine, IWV. It comprises the entire disease causing virus which is inactivated physically by heat or chemically. It has several antigenic parts to the host and can induce diverse immunologic responses against the pathogen. So this is how it works. And currently, there are 3 inactivated wall virus vaccine which has reached to phase 1 and 2 clinical trial. One is Beijing Institute of Biological Product, another is Sinovac company, and third is the most notorious place, Wuhan Institute of Biological Products. So these are the people who are working on that, and let us see what happens to this vaccine research. The third is the subunit vaccine, which contains the pathogenic derived proteins, antigens, with immunogenicity that can elicit the host immune system. And many research institutes have initiated this vaccine, and they are using the spike glycoprotein, which you have seen in the first picture, and its fragments also. So a company known as Novavax developed the candidate vaccine based on S protein. Clover Biopharmaceutical is another company, and they are developing a trimer vaccine, S trimer, by using its patented technology. And this is how the new things are coming. In monkeys, recombinant RBD protein successfully reduced the viral load in lungs and oropharynx, and to prevent this pneumonia. So this vaccines details we'll see later on, but this is the third type of vaccines, which is known as subunit vaccine. Now you can see that there is a host of companies and developers who are working all over world, day and night, by the biochemists, by the scientists, by the vaccine experts, by the technology developers, and various platforms, which are protein subunits, are on one platform, and you can see the classification of various protein subunit: Drosophila, S2 insect cell expression system, S protein, S trimer, we have already seen, again S protein, then S1 or RBD protein, adjuvant microsphere peptide, full length spike, S protein, and these are the companies: Generex, Epivax, Clover Pharmaceutical, Baylor College of Medicine, University of Queensland, and GlaxoSmithKline, then University of Saskatchewan in Canada, Johnson and Johnson, and many others. So they are developing these proteins and subunits, and viral challenges are taken up. The fourth important is the mRNA-based vaccines. When you have to develop the mRNA vaccine, there are 6 steps which are involved in this, and screening of antigen. Then you have to go for the optimization of the sequences. Then you have to go for modified nucleotide screening. Then delivery system optimization take place. Evaluation safety is studied, and then immune response is taken up. So a company which is in Boston, and its name is Moderna, as well as US based government institute, National Institute of Infected Diseases, they have launched the phase 1 clinical trial. And Bluebird Biopharmaceutical Company is also developing mRNA vaccine candidate using 2 different approaches. One is to use the mRNA to express the virus's protein and RBD domain, and the second is to express the virus-like particles in vivo. So Duke and NUS Medical School and this company, they are partnering this. Another this thing is a BioNTech and the other company, and the known company is Pfizer. They are collaborating and co-developing RNA-based vaccine candidate, which is known as BNT162. This is also a very recent development, and therefore I wanted to show you. The table second is also showing the vaccine candidate which are based on RNA platform, and this is according to WHO data, and where Moderna and other things we have already discussed. Imperial College of London, Pfizer, and China company, and Tongji University, they are the collaborators. Even Shanghai and Fudan University is collaborating. The fifth part is of vaccine is DNA vaccine, and DNA vaccine have a plasmid into which a particular gene is incorporated, and that encodes the antigens which identified from the pathogenic microorganism. So we are taking the bacterium plasmid, carrying the desired gene, delivered into the host, and translated the antigenic protein, which will stimulate the immune system normally activated by the pathogenic microorganism. So instead of pathogenic microorganism, it is the DNA vaccine. Bacterial plasmid will take call for this, and as on today, currently several pharmaceutical company has advanced DNA platform for discovery, and more DB candidates are working now, and the to prevent the attachment to the human ACE2 receptor itself, a receptor for the viral entry, because it is the first step. If you can counter that, nothing better than that. So there are two vaccines which are under development. Inovio Pharmaceuticals has developed in the name of INO 4800, and Techies and Applied DNA Sciences, and Avix and Zydus Cadila. So Zydus is a partner of developing a candidate by DNA vaccine. So you can see that the out of 5, Zydus Cadila, a company in our Gujarat, a flagship company of India, is taking care by developing the vaccine through the DNA vaccine platform and with collaborating with the others. The 6th is the viral vector-based vaccine, and you can see that the modified virus is utilized in this. Adenovirus is there, modified vaccinia virus, Ankara are the most common virus vectors in vaccine development. And there are companies and institutions, CanSino Biologicals, Beijing Institute of Biotechnology, Chen Y Group is also developing, Johnson & Johnson company. And there is a company which is known as Shenzhen Genoimmune Medical Institute. It is also developing 2 vaccines, and the work is going on. This is how the third table is showing the vaccine development done by using non-replicating virus platforms according to WHO as April 20. We are awaiting for the next results, and maybe the stages of clinical evaluation during this few months from phase 2, phase 1, preclinical, it may have advanced to a certain extent, but my idea is to show you the various non-replicating virus vectors which are used in the type of candidate vaccines. Who are the developers? You can just see that the Zydus Cadila is shown here, University of Oxford is shown here. Zydus Cadila is also working on this platform, and there are 2 candidates, NCT 033999578 and NCT 03615911. And this is the NCT where this is Shenzhen is working, and this is also the code which are given to this vaccine. The 7th type of vaccine, it is the virus-like particles. It is not virus, but it is the virus-like particles. They are the hollow core virus particles formed by the viral structural component with self-assembly character into nanostructure. And this VLP, it is shortly known as VLP, vaccine produced for greater than 30 different viruses. And GSK has successfully developed hepatitis B virus, Cervarix, that is human papilloma virus, and Merck has Recombivax hepatitis B virus platform, and also Gardasil, that is human papilloma virus. The receptor binding domain based on VLP platform vaccine candidate is derived from the cucumber mosaic virus, and currently it is under development. VLP can easily generated in a low containment production environment, and that is one of the plus point of this virus platform is used and developed. This is the latest situation of all virus vaccines which we have discussed so far. So it is the September 17 data. I couldn't get the updated data of last few days, but you can see that the University of Oxford and AstraZeneca, Gamaleya Research Institute, Janssen, Sinovac, Wuhan, Moderna, Novavax, Fosun Pharma and Pfizer, and this one. So these are the 12. Next, 13 to 24, where the Sanofi company is also one of this thing. Osaka University, Inovio we have already seen, and Kazakhstan Republic is also working in this. And these are the other platforms where, and Clover Biopharmaceuticals and GSK, and these are the type of vaccine candidates, and these are the vaccine platform. We have discussed all platforms very systematically, so that any first time learner can also understand these processes of development. This is the 25th to 36 molecules and development, and you can just see that the University of Queensland development, Cuba is also working on that, Sichuan University, Petersburg, and Imperial College London, and People's Liberation Army Academy, Military Academy of China is also working on this virus, and very famous universities like University of Queensland. So these are the type of vaccines we are helping. And now what we can look at, the development of many of the next generation platforms described here has so far been driven mainly by their potential use in the cancer therapy. COVID-19 pandemic has a fast-tracked their development as vaccine platform for emerging viruses. If current prediction becomes reality, then first vaccine against COVID-19 will be licensed within a year. These licensed vaccines are likely to include some of the next generation platforms described here. This in itself will be a major public health achievement, yet will simultaneously result in a permanent change to the vaccine platform landscape altogether, and an increased vaccine manufacturing capacity for this novel platform. You are aware that the government of India, as well as the worldwide scientists and governments and companies, they are looking at the large-scale manufacturing facilities which are built in India, and they are looking upon that, and ultimately the facility in India is looking upon their basic requirements from the glass bottles to the other requirement when the vaccine development platform and technology comes to India, how this technology go into the larger scale, and it is reached to the hospitals for the treatment of the patients as early as possible. So in this lecture, I just want to highlight the role of chemist, and when the role of chemist, biochemist, people in vaccine platform, and particularly the training in the immunology and the scientific society at large, working in the world community and various research universities, pharma companies, smaller pharma companies, smaller and smart and innovative pharma companies are playing collaboratively a very important role in development of vaccine. It is not a task of a single government, a single agency, a single institute, or a single-minded group of scientists. The collective effort from the leaders in technology, in science, in immunology, in biochemistry, in physiology, in allied science, when they will come together, then the things will happen and it will give the freedom to all of us as a human being to share this knowledge of immunology and vaccine to the world community, as in past the humanity has given several vaccines, and due to these vaccines we know that we have totally eradicated many diseases which were prevalent in global community as well as in India before and after independence. So this is a very important slide, and how immunology society can play an important role. But I think that it is also true that how chemist, biochemist, immunologist and researcher, bioinformatic persons, drug design people, combinatorial chemistry people can work together to come to a newer molecules, better molecules, and they can study the older molecules which were failed, which are patented once upon a time, which are now off patented, and if this literature is resurveyed, revisited, and new strategies for small molecules can also be attracted, and young researchers will take up a call that why not we can come back to the old molecules which were failed molecules, but they were better, they were toxic and they were failed, but we can utilize it as a repurpose, we can reframe their constitution, we can reduce the toxicity, and we can increase the potency of these molecules. So my take-home message to all of us that let us work collectively in group, understand the requirement of research and how the step-by-step research in a long way, like a 50 or 100 compartment train is working, and there is a what we call it, the driver seat, as well as in the backyard there is a seat for the guard. So we have to reassemble the chemist, biochemist, and all other branches of science to take up this challenge, because as on today we know that there is no complete solution of COVID-19, a very mischievous virus which is changing its genetic platform and its genetic codes at different location differently, and therefore the challenge is much more than we can foresee on the surface. So with this concluding remark, I wish all of you a very challenging research career as well as your academic career. Thank you all. Thank you for inviting me on this occasion, and I'm very happy that I have shared my few information with you people. Thank you.